All right, good morning, guys. Last week, I posted a video where we talked about the nervous system, the structure of the nervous system, and we had talked about the different cells that were present there. And throughout ANP1 and here at the very beginning of ANP2, I've said things about how a nerve made reference to a nerve impulse. And I said it's kind of, not exactly, but it's a little bit like electricity, because it does involve ions as opposed to electricity involving electrons, the flow of electrons from one location to another. Well, it's not quite as simple as that, but we are going to talk about ions moving across the cell membrane. So throughout the semester, we've talked about cells, and people think, why do I have to know about cells? I'm studying anatomy, human anatomy. Well, the reason for that is that you have to understand about cells in order to understand what's going on when we talk about a nerve impulse. And we're going to be making reference to the location for different ions. And so we're going to start out here at the very beginning, and we're going to talk about a neuron, and we're going to talk about the distribution of ions inside and outside of that neuron. So what we're talking about is cell membrane potential, in particular, what we call the resting cell membrane potential. Now, what is potential? Potential is the difference in charge from two different locations. So if you think about this, you might think of a battery. And so you might think, a 9-volt battery, it has those two different kinds of tops on it. And the difference from one location to another is 9 volts. That's the difference in the concentration of electrons in the case of here. Negative ions electrons, which are negative, versus the positive pole. When we talk about resting membrane potential in the neuron, what we're talking about is the difference in the charge on the inside versus the outside of the cell. The intracellular fluid versus the extracellular fluid. That's the fluid outside. You might remember that I had mentioned something called the sodium-potassium pump. And we had said there's a huge amount of metabolism, the burning use of energy that goes into pumping ions. And so what it's going to be doing is it's going to be pumping sodium out, while at the same time it is bringing potassium in. And so as a result of this, and so we have the sodium-potassium pump. And then in the cell membrane, we have some channels as well. So the sodium-potassium pump is active transport. So it's pumping. But then we have some what we call leakage channels. And remember, we had said a leakage channel or a channel protein it's kind of like a hole in the cell membrane that lets one kind of ion or water, one kind of thing to pass through. And it can go in or out. So we have leakage channels as well. So we have sodium that is actively being pumped outside of the cell, potassium that simultaneously, as it's pumping sodium out, it's pulling potassium in. And then we have these channels that allow stuff to leak in or out We have sodium leakage channels and we have potassium leakage channels as well. And even chloride. So we have these channels that allow stuff to move by diffusion as well. And so what we end up having, and we have one other major and very important component. We have proteins on the inside of a cell. You know that. We had talked about transcription and translation last semester. And so what we had said, what is that? That's all about making proteins. Well, it ends up those amino acids that are the building blocks of a protein. Some of them have R group. Remember R groups? Yeah. You said it's a bad memory. I don't want to think about it. Well, anyway, think about it a little bit more. And those R groups, it ends up, there are more of those that are negative than there are positive charged ones. So on the inside of the cell, we have some proteins. They're way too big to go through a leakage channel or to be pumped. So we're not talking about them leaving, but what we're talking about is the potential, the difference in the charge inside the cell versus outside the cell. And so we have these ions that are flowing around, they're being pumped and they're leaking, and then we have the R groups on the proteins, and all of it in this sort of a mess of materials, we're going to end up with a potential, a charge. Now, when I talked about the potential of that square battery, like you put in your fire alarm, we had said 9 volts, right? Well, it ends up with all of this pumping and leaking and so forth, we don't have anywhere near that big a difference between the inside and the outside of the cell. We have millivolt differences. Now, what is a millivolt? A millivolt is 1,000th of a volt. So when we say, okay, when we have a nerve cell that's just sitting there resting, doing nothing, which doesn't ever happen, but experimentally they can set up this situation. And we know that the resting membrane potential, this is important, it's on slide number 4, is minus 70 millivolts. So it means it's slightly negative, minus, on the inside versus the outside. There will be, just so that you understand, when we're talking about that surface of a cell. So here what we're looking at is a leakage channel. Here we're looking at the sodium-potassium pump and another leakage channel here. It ends up there's even more different kinds of structures there in the cell membrane. There's some that have like a top that can open. So if we have say that's the channel, you can see the hole. Hopefully you can see the hole through which something could pass. All right, but if I put my thumb over the top, you can't get through it, right? So there's something called gated channels, voltage and chemical gated channels. And they're channels that can open or close. So keep that in mind as we talk, as we move forward with this. So we're going to talk about how these gated channels can open and close in response to a change in voltage. And so the very first thing that we've learned about, in addition to these channels and active transport molecules in the surface of the cell membrane, is that when the nerve cell is not communicating with anyone else, just doing its thing, hanging out, it's at minus 70 millivolts. So what do we call minus 70 millivolts? The resting membrane potential. You will see that again. Make sure you remember that. It's minus 70. And make sure you, more importantly, make sure you understand why it is. It's because you're pumping stuff out at the same time you're pumping stuff in, and it's leaking. The sodium that's being pumped out is gradually leaking in, and the potassium that's being pumped in is gradually leaking out. But overall, we're at this minus 70 millivolts, which is the resting membrane potential. So now, why am I talking about this? The reason I'm talking about this is because we're going to talk about a nerve impulse, and it's going to involve, those voltage and chemical gated channels. Aha, something to think about in the next little presentation.